when I go to parties, it doesn't usually take very long for people to find out that I'm a scientist and I study sex. And then I get asked questions. And the questions usually have a very particular format. They start with the phrase, "A friend told me," and then they end with the phrase, "Is this true?" And most of the time, I'm glad to say that I can answer them, but sometimes I have to say, "I'm really sorry, but I don't know because I'm not that kind of a doctor." That is, I'm not a clinician. I'm a comparative biologist who studies anatomy. And my job is to look at lots of different species of animals and try to figure out how their tissues and organs work when everything's going right, rather than trying to figure out how to fix things when they go wrong, like so many of you. And what I do is I look for similarities and differences in the solutions that they've evolved for fundamental biological problems. So today I'm here to argue that this is not just, well, or not at all, an esoteric ivory tower activity that we find in our universities, but that broad study across species, tissue types, and organ systems can produce insights that have direct implications for human health. And this is true both of my recent project on sex differences in the brain and my more mature work on the anatomy and function of penises. And now you know why I'm fun at parties. So today, I'm going to give you an example drawn from my penis study to show you how knowledge drawn from studies of one organ system provided insights into a very different one. Now, I'm sure, as everyone in this audience already knows, I did have to explain it to my 9-year-old last week. Penises are structures that transfer sperm from one individual to another, and the slide behind me barely scratches the surface of how widespread they are in animals. There's an enormous amount of anatomical variation. You find muscular tubes, modified legs, modified fins, as well as the mammalian, fleshy, inflatable cylinder that we're all familiar with. At least half of you are. And I think we see this tremendous variation because it's a really effective solution to a very basic biological problem, and that is getting sperm in a position to meet up with eggs and form zygotes. Now, the penis isn't actually required for internal fertilization, but when internal fertilization evolves, penises often follow. And the question I get when I start talking about this most often is, what made you interested in this subject? And the answer is skeletons. You wouldn't think that skeletons and penises have very much to do with one another. And that's because we tend to think of skeletons as stiff lever systems that produce speed or power. And my first forays into biological research, doing dinosaur paleontology as an undergraduate, were really squarely in that realm. But when I went to graduate school to study biomechanics, I really wanted to find a dissertation project that would expand our knowledge of skeletal function. I tried a bunch of different stuff. A lot of it didn't pan out. But then one day I started thinking about the mammalian penis. And it's really an odd sort of structure. Before it can be used for internal fertilization, its mechanical behavior has to change in a really dramatic fashion. Most of the time, it's a flexible organ. It's easy to bend. But before it's brought into use during copulation, it has to become rigid. It has to become difficult to bend. And moreover, it has to work. A reproductive system that fails to function, produces an individual that has no offspring, and that individual is then kicked out of the gene pool. And so I thought, here's a problem that just cries out for a skeletal system. Not one like this one, but one like this one. Because functionally, a skeleton is any system that supports tissue and transmits forces. And I already knew that animals like this earthworm, indeed most animals, don't support their tissues by draping them over bones. Instead, they're more like reinforced water balloons. They use a skeleton that we call a hydrostatic skeleton. And a hydrostatic skeleton uses two elements. The skeletal support comes from an interaction between a pressurized fluid and a surrounding wall of tissue that's held in tension and reinforced with fibrous proteins. And the interaction is crucial. Without both elements, you have no support. If you have fluid with no wall to surround it and keep pressure up, you have a puddle. And if you have just the wall with no fluid inside of it to put the wall in tension, you've got a little wet rag. When you look at a penis in cross-section, It has a lot of the hallmarks of a hydrostatic skeleton. It has a central space of spongy erectile tissue that fills with fluid, in this case blood, surrounded by a wall of tissue that's rich in a stiff structural protein called collagen. But at the time, when I started this project, the best explanation I could find for penile erection was that the wall surrounded these spongy tissues, and the spongy tissues filled with blood, and pressure rose, and voila, it became erect. And that explained to me expansion. Makes sense, more fluid, you get tissues that expand. But it didn't actually explain erection, because there was no mechanism in this explanation for making the structure hard to bend. And no one had systematically looked at the wall tissue, so I thought, wall tissue is important in skeletons, it has to be part of the explanation. And this was the point at which my graduate advisor said, whoa, hold on, slow down, because after about 6 months of me talking about this, I think he finally figured out that I was really serious about the penis thing. So he sat me down and he warned me. He was like, be careful going down this path. I'm not sure this project's going to pan out. Because he was afraid I was walking into a trap. I was taking on a socially embarrassing question with an answer that he thought might not be particularly interesting. And that was because every hydrostatic skeleton that we had found in nature to that point had the same basic elements. It had the central fluid, it had the surrounding wall, and the reinforcing fibers in the wall were arranged in crossed helices around the long axis of the skeleton. So the image behind me shows a piece of tissue in one of these cross-helical skeletons, cut so that you're looking at the surface of the wall. The arrow shows you the long axis, and you can see 2 layers of fibers, one in blue and one in yellow, arranged in left-handed and right-handed angles. And if you weren't just looking at a little section of the fibers, those fibers would be going in helices around the long axis of the skeleton, something like a Chinese finger trap, where you stick your fingers in and they get stuck. And these skeletons have a particular set of behaviors, which I'm going to demonstrate in a film. It's a model skeleton that I made out of a piece of cloth that I wrapped around an inflated balloon. The cloth's cut on the bias, so you can see that the fibers wrap in helices, and those fibers can reorient as the skeleton moves, which means the skeleton is flexible, it lengthens, shortens, and bends really easily in response to internal or external forces. Now, my advisor's concern was, what if the penile wall tissue is just the same as any other hydrostatic skeleton? What are you going to contribute? What new thing are you contributing to our knowledge of biology? And I thought, yeah, he does have a really good point here. So I spent a long, long time thinking about it. And one thing kept bothering me, and that's, when they're functioning, penises don't wiggle. So something interesting had to be going on. So I collected the wall, I went ahead, collected wall tissue, prepared it so it was erect, sectioned it, put it on slides, and then stuck it under the microscope to have a look, fully expecting to see crossed helices of collagen of some variety. But instead, I saw this. Well, there's an outer layer, an inner layer. The arrow still shows you the long axis of the skeleton. I was really surprised at this. Everyone I showed it was really surprised at this. Why was everyone surprised at this? That's because we knew, theoretically, that there was another way of arranging fibers in a hydrostatic skeleton. And that was with fibers at 0 degrees and 90 degrees to the long axis of the structure. The thing is, no one had ever seen it before in nature. And now I was looking at one. Those fibers in that particular orientation give the skeleton a very, very different behavior. I'm going to show you a model made out of exactly the same materials. So it'll be made of the same cotton cloth, same balloon, same internal pressure, but the only difference is that the fibers are arranged differently. And you'll see that unlike the cross-helical model, this model resists extension and contraction and resists bending. Now, what that tells us is that wall tissues are doing so much more than just covering the vascular tissues. They're an integral part of the penile skeleton. If the wall around the erectile tissue wasn't there, it wasn't reinforced in this way, the shape would change, but the inflated penis would not resist bending, and erection simply wouldn't work. It's an observation with obvious medical applications in humans as well. But it's also relevant in a broad sense, I think, to the design of prosthetics, soft robots, basically anything where changes of shape and stiffness are important. So, to sum up, 20 years ago, I had a college advisor tell me when I went to the college and said, I'm kind of interested in anatomy, they said, anatomy is a dead science. He couldn't have been more wrong. I really believe that we still have a lot to learn about the normal structure and function of our bodies, not just about its genetics and molecular biology, but up here in the meat end of the scale. We've got limits on our time. We often focus on one disease, one model, one problem. But my experience suggests that we should take the time to apply ideas broadly between systems and just see where it takes us. After all, if ideas about invertebrate skeletons can give us insights about mammalian reproductive systems, there could be lots of other wild and productive connections lurking out there just waiting to be found. Thank you.